Dear students, today we are going to discuss about the Mendalism, genetical study of Mendel. You know, he is the monk, he is not a scientist, he is not a science student, but he became the father of genetics. He became the father of biology, one of the biology branch. His natural habitat, actually he is doing as a gardener in the garden. In his regular observation, he observes so many varieties of the flowers, so many varieties of the plant, each plant having the different type of the flowers, different type of the fruit, seed. Some of them are coming in the middle, some of them are coming at the tip of the plant. This variation he has observed years together. After observation of years together, he decided to study about these plants and hybridization of the plants. Actually, he is the monk, he is the mathematician, he studied the mathematics, he is worked in the Natural History Museum. His father is a normal farmer, he came from a poor family, and he is very much interested on the plant, especially in the hybridization of the plant. Why? Is routinely is going in the garden, routinely is seeing the so many varieties of the plant and flowers. That is why, though he is not the biologist, he has very much interest to study of the plants. That is why he has studied first time he used the mathematics, probability, correlation, correlation, coefficiency. These all mathematical problems are involved in the biology. In the biological study, use the mathematics. You know, the genes, about the study about the gene, actually as like a biology, zoology, we have to study the genealogy, but we are not studying it as a genealogy. Genetics means study about the genes which are there, how heredity and variation, how the heredity, how the genetic characters are going from one generation to other generation, their offsprings. Some of them they having the variation. You know, some of the people, they what they said, oh, he is look like his father, he is look like his mother, he is look like his grandmother, he is look like his grandfather. But some of them they call, hey, his children is whether the father is there or not, father is or not. Why? He is a white and very high personality, but his children may be very black and very shouted. That variation is happening, generally it happens, sometimes very less amount of the variation is happening. Not only the heredity, pure genetic characters, variations also coming from parents, because of the condensation, because of the linkage, because of the crossing over, sometimes variation is happening. Studying about these two, heredity and variation, these two is commonly called as genetics. We are using the mathematics in the gene study, that is why we are calling it as genetics, not the genealogy, genetics. Okay. Here he has studied lot, and especially he has taken single character, especially the, you know, the as we are studied in the chromosome, each plant or each living organism having the specific number of the chromosomes. As in our human being, we having the 23 sets of the chromosome, 22 sets of the chromosome are autosomes, one set is allosome, sex chromosome. As like in the botany plant, that means Pisum sativum, botany plant, 7 sets of the chromosome, that means 14 chromosomes are present. That is why it having the 7 different characteristics. He has given the name it as trait, factor. Earlier he has not given the name it as a gene name given by Johansson, but at the time of the study, Mendel study, he has given it name it as factor. One factor it is causing for the either tall or short, either flowering red or white, either flowering at the tip or at the nodal face. These all are the characteristics that are causing by that special traits or causing by special genes. That special traits we calling it as traits, now we calling it as genes. What the Mendel said it as a traits or factors, but now we are calling it as a genes. That genes where the genes are present in the chromosome, that we call as locus. You know, the one set of the chromosome, that means two chromosome, it shows the same characteristics, it is present in the same locus, same space, that we call as alleles. Alleloia, not alleloia, alleles. That means two chromosome or pair of the chromosome which are there in the same place and which show the same characteristics. Suppose height, some set of the chromosome is deciding the height factor. That height factor deciding the 2, that means pair of the chromosome, pair of the genes traits are there. That pair of the genes we call as alleles. What the alleles it is showing phenotypically, that we call allelomorph. That all he has studied. 7 characters he has studied individually. He has taken one specific character set, that means monohybrid, and 2 characters he has taken dihybrid, and he has not gone though above. That is why he is very perfect. He could able to initialize, he become the father of genetics, because he has chosen the best plant, very small life cycle plant. Easily we can self pollinate, easily we can cross pollinate, easily we can study the pea plants in more number, luxuriantly grown in the garden, and easily we can cross pollinate, and easily we can observe the flower, seed, fruit without any waste of the time, without any waste of the much force, much time, easily absorbed. Why? He is in the garden. That is why he has taken the pea plant and he observed lot. He has utilized the mathematics. He has Punnett square box. He utilized the Punnett square box. You know, Sudoku. What is the game changer? What that we can change, modify into one set, one color at one side, one color at one side, like that. The earlier in that 19th century also Sudoku like game, that the Punnett square, with that we can easily observe, easily identify, easily categorize the type of the genome, type of the characters, that all we can easily observe. You know, that traits name given by traits name given as a gene by Batson. You know that, but he has studied lot, but his study is not recognized anywhere. His study is not recognized anywhere. He studied his own. He proposed the hybridization experimental theory of hybridization and experimentation on plant hybrids. He has written the book, but later after his death, 3 different scientists from 3 different states, 3 different countries, from Hugo DeVries and Carl Correns and Sermar. These 3 scientists again re-studied, reintroduced, re-research they have done and identified the Mendel work is the perfect work. Mendel has done the great work that is very useful to study, to know about the genetic structure, heredity and variation. Then the 20th century, at the start of the 20th century, it started the genetic study after his death. Though he is initiated, on his name they proposed the Mendel theories. The Mendel theories are there, 3 Mendel theories: law of dominance, law of segregation, law of independence. 3 different laws they propose on based upon the Mendel laws, Mendel theories. Okay, you know the where the gamete it having the haploid set of the chromosome, that we calling it as gene. That gene where it is present in the chromosome, that is called as locus. The gene is the hereditary material, that is the functional unit of the heredity. Okay. And you know the one gene, one enzyme theory proposed by Avery, McLeod and McCutch. Avery, McLeod and McCutch, these 3 scientists studied, based upon their study, they proposed the DNA as a genetic material, not the protein. And you know Beadle and Tatum have done the theory, studied after this theory one gene, one enzyme. That means each gene that produce specific type of the enzyme, specific protein. The gene is universal. Gene can produce, DNA can produce same type of the production, even in it is in the microorganism or in the large elephant. That is why it is a universal, it is a unique, it having the specificity, it having the polynomicity, it having the very highest accuracy. So that is why we could able to produce our hormones, our vitamins, our enzymes, which we are producing in our human body, we could able to produce by using the microorganism. Without killing any living organism, we could able to synthesize our insulin. Otherwise, earlier we could kill the horse and we extract the insulin. But now, because of the study, all gene study, genome study, gene mapping, chromosome mapping, we could able to synthesize the insulin artificially by using the microorganism. Instead of production, it produced in the human beings. But whatever the gene which we inserted into the microorganism, that microorganism also producing the same. That is the capacity of the genetics. You know, there is a lot of terms are there in the genetic study. Heredity, you know, the heredity means whatever the traits, character which came from the parents to the offsprings, that is heredity. And you know the monohybrid, that means only if you take only one trait, one single character differentiation, height, tall plant and short plant, pink color flowers and white color flowers, only one trait, different trait to different characteristics of the same height, short, tall or short, pink or white, and round or wrinkle, folded, like that. You know, alleles, I already told you, the two forms of the genes which having the same characteristics. And you know, the Mendel studied only two, monohybrid and dihybrid. Okay. And he has studied, he could able to know the dominant characters and recessive character. Whenever they do the hybridization in between the two plants, which character is coming to the next generation, that is the dominant character. If we cross between the tall and short plant, next generation only tall plant will come, not the short plant. Why? Because of the dominant. Which one, which allele, which gene it has in the dominant character, the dominant the remaining gene, the dominant the remaining gene. That is why always the dominant character will express in the first generation. But in the next generation there is a chance, 25% of the chance to come the tall, maybe some of the short will come, 3:1, 1:2:1. Which one, which gene, which characteristic gene that is suppressing, that is called as recessive. After hybridization, whatever the trait, whatever the characteristics which are visible to us, that is phenotype, which are not visible, but it is genotypically it is present in the plant inside, that is genotype, genetic state, genetic character, DNA characters, that is genotype. And you know, if the two chromosomes of the same type, suppose capital T, small t, or capital R, same two sets, that means two pair of the chromosome which showing the same characteristic, that is homozygous, one from father, one from mother, though it is the same, that is homozygous. If it is a different, one is capital T, one is small t, that is heterozygous. Homozygous, heterozygous. Heterozygous means hybrids. Here you can see the Mendel experiment, how he has done the experiment, just 1 minute video. In the Mendel in the garden, he has studied on the pea plants. You see how he has done the work. He collected different varieties of the plant, different variations, different varieties of the plant here. You see the pink color flowers and white color flowers, and the flowers which are at the typical, flowers that are the axial. You see what he has done, he is exchanging the pollen material from one to other. He is spraying the pollen upon the pistil, stigma. See, you see, and then whatever the seeds he collected and he has shown in the next generation, what he has observed, all the majority of the number are pink color. He has done, what he has done here, you see emasculation, that means removing of the male part from the flower, so that whatever the desired plant we want, that type of the desired plant we can. You see the pollens from the short plant or pollen from the white flower plant, and stigma female will become the pink color. They two attached, so that they crossed hybrid, they crossed the between the two plants, and then they got they collected the seeds, and the collected seeds again they shown the next generation F2 generation, parental to first generation F1 generation and the F2 generation. In the F1 generation all will become the pink, but whereas in the F2 generation some of them are white. You see the white color happen. Why the white color behind it? So there is some recessive genes also present that express in some time. That is why these all shown recessive character also phenotypically that expressed. You see the Mendel, he has taken 7 different traits, 7 different characteristics, each characteristics having the one set of the gene. That is why you know the Pisum sativum, this one pea plant having the 7 set of the chromosome, that means 14 chromosome, each chromosome having the specificity. Suppose flower color, pink color and white color, flower production, flower at the typical, flower at the axial, and you see the seed yellow color or green color, and the seed coat you see it is rounded or folded, winkled, folded or rounded, and you can see the fruit, how the fruit is fully swollen or bulged or folded, and you can see the fruit, ripened fruit, how it is, green color or yellow color, how it is. The variation is there in each character, differentiation is there. Like that he has taken each character one time, that is monohybridation, one character he has taken. Like that 7 character, 7 different plant he has taken. He observed whether it is a phenotypically correct or not, whether it is homozygous or heterozygous, whether it is our same set of the chromosomes are present or different set of the chromosomes are present. That all he has observed. With this observation, you know the why the pea plant, what is the reason behind the success of the Mendel. He has taken the very simple plant, it having the vigorous growth, and we can do the self fertilization, it take the self fertilization, and it produces more number of the upsprings within the short span of the time, and we can isolate easily with the 7 traits, and easy to breed. They reproduce sexually and have 2 distinct male and female sex gametes we can easily identify, and you know the cross pollination is very easy, very feasible, and also lot of variation is there in between the 2 traits, and reproduction also very easy, smooth type of the reproduction, and control the mating, we can control the mating of the male and female, and we also can short the plant having the very short lifespan, so that we can study the more and more generations, and large number of the offsprings we can study within the short span of the time, and also convenience for the breeding, very easy. You know here he has done the monohybrid cross first, he has taken only one single character to study. In the single character, what is the variation, whether the variation is there or not. They tried, he has taken tall plant and short plant, he hybrided between the 2, tall plant and short plant. Short plant pollen he pour on the tall plant pistil, so that the fertilization is taken place, and then the first generation all the plants are tall, and then he astonished, why only the tall, why not the short. Again for the observation, again he has done the next generation study. In the next generation study, he could able to study, we could able to identify. Here you can see the next character, flower character also only one character he has taken, pink color flower, white color flower, whereas he has taken the first generation all the flowers are pink, that is dominant. Which character is dominant? That expressed in the first generation. Which character is recessive? Which character is recessive, suppressive? That is not expressed outside. That is phenotypically we can't see it. That is the recessive. Here you can see the dominant one. Which one is the dominant? that expressed in the first generation. You can see in the 7 characteristics he observed, he has done the cross pollination, he has done the hybridization in the 7 different, that means seed size, whether it is round or winkle, and cotyledon, how the cotyledon is yellow or green, and the pod shape, pod how it is bulged or winkle, or seed is rounded or winkle, seed yellow or green. The 7 flower at the typical or at the axial, 7 different characters he has taken. All the characters almost it is in the F2 generation he came 3:1, 3:1, that means 3 dominant characteristic chromosomes having plants and 1 recessive characteristic plants he observed. You can see here what they used, that is Punnett square box, that prepared by Punnett, that is Punnett square box. In the Punnett square box we use the gametes in one x axis side and y axis side, from female x axis side and male from y axis side. The both we could cross with that. Suppose in the first box capital T with the capital T, and in the second box capital T and small t, and in here in the second box capital T and small t. Here in the fourth box small t, here also small t, here also small t. So that all the small recessive, there is no dominant character here, there is no dominant trait, there is no dominant gene. That is why here the recessive character which are expressed. That is why one white flower or one short plant is germinated from the pure pink color. Well, for this study he has done the back cross and test cross for the observation whether the we are taken is looking like a tall, whether it is a homozygous perfect tall or it having the short character also inside. To observe it, to identify it, he has done the back cross and test cross. Back cross means whatever the F1 generation, first generation plants, he hybridized with the parental plant, hybridized with the parental dominant species plants. So that here you can see the capital T dominant one side, and that is another one that I will show you. Here you can see the dominance where there is a, if you see the tall plants and short plant, whenever they hybridized with the same, automatically in the next generation 3:1. If we go for the genetic genotype that are capital T 1 and capital T small t 2 and small t 1, 1:2:1, that is the genotypic ratio, genetic characteristics of the plant. And then you can see in the test cross, the test cross are there, you see here back cross and test cross, how it is. Here whatever the F1 generation, it having the capital T and small t, that he hybrid with the dominant characteristic plant. Then what happened? All the plants are dominant, all the plants are tall. We could not able to see whether it is homozygous or heterozygous. We are having the same set of the chromosome or different set of the chromosome. But in the test cross, we can easily identify that test cross is what it is, whatever the F1 generation, by hybridizing with the 2 different parental characteristics, that F1 generation is mating with recessive, that means not dominated, recessive, that means small t, with the small t, if we mate, the what happen, half of the plants are tall, half of the plants are short, that means 1:1, that means 50% of the dominant gene is there in the chromosome, 50% of recessive gene is there. So that there is a heterozygous, father gene and mother gene are different. That is why heterozygous type of the plants came. Even in the dihybrid also, you could able to identify 4 different type of the seeds: round yellow, round green, and round winkle, that means yellow winkle and green winkle. These are 4 types you could able to identify, 4 type, each one. Based upon that, you could able to know whether what are the plant we are seeing, that is perfect having the same characteristics or it may having the jumbling characteristics, that he has observed. And the next one is dihybrid cross. That means he has taken two characteristics, two different characteristics. Suppose seed, if you take the seed, seed color yellow, seed color green, and the seed shape round or folded. Two characteristics he has taken. He hybridized in between the two different characteristics plant, two different traits. The two different trait characteristics he made, so that whatever the next generation, how it is coming, he observed with this observation he could able to identify gametes having the purity or not, whether the gamete are pure or there may be any disjunctions and maybe any another part may be added, that he could able to identify. That means round R, whether it is a dominant character, that we use capital letter, capital R, and for the recessive character we use small r, that means folded. And the yellow color is a dominant character, that is why capital Y we use, and the green color is a recessive character, that is why we use small y. In between the two different traits, they hybridize. So, in the hybridization, first generation he could get all the round and yellow seeded plants, round and yellow seeded plants. There is no green folded plants. The round and yellow plants again he mated with the same, that means he has done the self pollination. With the self pollination he could get the four different traits, that means round yellow color could able to mate with the round and also could able to meet the winkle. That is why capital Y also mating with, whenever the gamete formation, only one set of the chromosome will come. Of the two set capital Y, only one Y will come, and from the two capital R, one R will come. So, that capital Y can easily mate with the small r, capital Y can easily mate with the capital R. There is no condensation, there is no strict rules. They can mate with any others. They having that independence. That means independent assortment, law of segregation, law of purity. Based upon that, he proposed the three theories. Here you can see the what are there: yellow round and yellow folded, and green round and green folded. Four characteristics he has taken, and then he could able to find 16 various type of the plant, 16 various characteristics. Of that, what are the types are there? How many are round yellow? How many are round green? How many are folded yellow? How many are folded green? You see here 9. These 9 are round and yellow, dominant characteristics it is showing. And then you can see the remaining 3 in the second box and last second and here last second. These 3 are yellow and folded. Yellow and folded are there. And again the next 3 you can see these 3 are what are the green and round. Green it is round, and only one, that is parental characters, recessive character, that is what it is green folded. You could able to see 9:3:3:1 ratio, that is phenotypic ratio, how it is looking like. And whereas genotypic ratio, genetically there is a variation, capital Y, capital R, only one plant is there, pure yellow round, and heterozygous yellow round 2, and heterozygous yellow and homozygous round again 2, and then heterozygous yellow, heterozygous red round are 4, and homozygous yellow and homozygous winkle only 1, and homozygous heterozygous yellow and homozygous Winkel, that are 2, and again homozygous recessive, that means green, and heterozygous homozygous round, only 1, and then again homozygous green and heterozygous round. So, gene study is very very important to understand the level, to understand the characteristics, to understand the generation study, to understand their standard level, IQ level, that all is very very important. That is why we have Mendel, even is the Mark, we calling it as a father of genetics.